And the next question is, what were the 7 characters which Mendel selected for his experiments, and classify these characters into their dominant and the recessive forms? We know that Mendel had selected 7 pairs of contrasting characters, and there was every time a dominant character and a recessive character in each case. So the 7 pairs of contrasting characters are given as follows. He took the length of the stem, where there were 2 possibilities, tall and dwarf, while the tall was dominant over the dwarf. The second one was position of the flower. Again, over here there were 2 possibilities, axial when it is present at an angle and terminal when it is present at the tip. The third one was color of the pod or the fruit. Over here the green colored pod is dominant over the yellow color. The next one is shape of the pod. Again, there were 2 possibilities, inflated, that means swollen, and constricted, where the margins were not entire. The next one was color of the seed coat, where colored was dominant, whereas white was recessive. He also took shape of the seed, where, as we had used in the dihybrid crosses, round versus wrinkled. While the round one was dominant, the wrinkled seed was a recessive character, that because it did not express itself in the F1 generation. And the last one is color of the cotyledon. Over here, yellow colored cotyledon is dominant over the green color. The next one is define the term allele. Now, the individual members of the gene are called alleles. So, alleles are basically the members of the gene. They occupy the same locus on the homologous chromosomes and control the same character but in different ways. For example, if tallness is a character, it is controlled by 2 alleles, capital T and small t. So when capital T and capital T are together, the character expressed is tallness. When small t are together, the character expressed is dwarfness, while when they are present in the heterozygous form, again it is expressed as tall, because tallness over here is dominant over dwarfness. The next one is, why is variation necessary? So before we understand the role of variation, we should understand what is variation. Variation is basically change in the traits or characteristics between individual of the same species. And why is it necessary? Because whenever there is a change, whenever a change takes place either due to evolution or mutation, then it leads to better adaptability to the surroundings. So variations are necessary. And the last one is, how many types of variations are there in a population? We know that variations are of 2 types. There is discontinuous variation and there is continuous variation. So discontinuous variation, in such kind of a variation, there will be clear-cut characters. Like in case of Mendel, there were tall pea plants and there were short pea plants. So there were clear-cut characters. There were no gradations, right? Whereas in case of continuous variation, there are gradations. There's no clear-cut character. No 2 characters are clear-cut, as in the human skin color, the human height. It follows. It has many phenotypes and they are like graded. So you see that the human skin color has approximately 64 kinds of phenotypes, ranging from Negroes to albinos and many intermediates. So continuous has got different gradations, different phenotypes. Also, in case of discontinuous, the discontinuous is not affected by the environment, while the continuous is affected by the environment. So these are the differences between discontinuous and continuous variation.